Hello and welcome to Sleepy Science Doc. Tonight, we wander into a world where petals unfold beneath the sun's first touch. A realm built not by machinery, but by the patient work of roots, reaching through soil and stems stretching toward light. Flowers hold secrets written in fragrance and color, stories that bloom across seasons in gardens that whisper with life. Here, beneath the gentle passage of clouds, entire kingdoms rise from the earth. Blossoms of impossible delicacy reach toward filtered sunlight. Sanctuaries form in hidden meadows, and every petal tells a tale of resilience, beauty, and the quiet dance between plants and the creatures that need them to survive. If these peaceful explorations bring you comfort, I gently invite you to subscribe, share your thoughts, or simply rest here for a while. Your presence helps others discover this tranquil corner of wonder. But for now, allow your shoulders to soften. Let the tension drift away from your neck like morning mist, evaporating in warm air. Breathe slowly, and let us begin this serene journey together. Some flower petals are so thin that light passes through them like stained glass. These delicate membranes possess a translucence that transforms sunlight into soft colored glows. When backlit by morning rays, certain poppy petals become luminous screens of orange and crimson, their veins visible as darker threads against the radiance. The thinness serves purposes beyond beauty, allowing maximum light capture for the reproductive structures hidden at the petal's base, while minimizing the energy required to construct such ephemeral tissues. Each petal exists for only days, sometimes mere hours. Yet during its brief life, it creates moments of transcendent loveliness. The fragility makes them vulnerable to wind and rain, yet their numbers provide strength. A single flower might bear dozens of these gossamer petals, and a meadow might hold millions, creating landscapes that shimmer with transmitted light. Standing in such a field feels like existing inside a lantern, surrounded by living color that breathes and moves with each passing breeze. Certain blossoms release their fragrance only when darkness falls. As daylight fades and shadows lengthen across the garden, some flowers undergo a quiet transformation. Structures that remained closed and scentless during the day suddenly open, releasing waves of perfume into the cooling air. Night-blooming jasmine fills tropical evenings with sweetness, so intense it seems to have physical weight. Moonflowers unfurl white trumpets that glow in starlight while exhaling vanilla-scented clouds. The timing connects to pollinators that fly in darkness, moths with tongues longer than your finger, bats that navigate by echolocation, beetles that crawl from hiding as temperatures drop. These night shift workers require different signals than daytime visitors, so the flowers adapt, saving their most potent scents for the hours when their specific partners are active. The transformation happens gradually, a slow opening that responds to dropping light levels and shifting air chemistry. By dawn, many of these nocturnal flowers close again, their petals folding inward to protect delicate reproductive parts from the scorching sun. Their fragrances fading like dreams upon waking. Some flowers open their petals in spirals that follow mathematical patterns found throughout nature. The unfurling happens according to sequences where each petal's position relates to the previous one by a specific angle, creating arrangements of perfect efficiency. No space is wasted, no overlap excessive. Roses demonstrate this principle beautifully, their petals nested in walls that satisfy both aesthetic and functional demands. The mathematical precision emerges not from conscious design, but from simple growth rules, applied consistently as the bud develops. Each new petal forms at the optimal angle to maximize its access to light while fitting into the limited space of the developing flower. The resulting spirals echo patterns seen in seashells, galaxies, and the arrangement of seeds in a sunflower head. This mathematical beauty exists whether humans observe it or not. A testament to the elegant solutions evolution discovers through countless generations of trial and refinement. When we find flowers beautiful, perhaps we're responding unconsciously to these underlying patterns, recognizing at some deep level the efficiency and balance they represent. Certain petals contain microscopic structures that create colors without pigments. Instead of relying on colored chemicals, some flowers build tiny ridges and valleys on their petal surfaces that interfere with light waves, bending and splitting them to produce iridescent hues. These structural colors shift depending on viewing angle, creating flowers that seem to change shade as you move around them. The effect is similar to oil on water or a butterfly wing, where the color comes from physics rather than chemistry. Some orchids use this technique to create ultraviolet patterns invisible to human eyes, but brilliant to the bees they're trying to attract. The microscopic architecture required for these effects is breathtakingly complex, with structures measured in nanometers, thousands of times smaller than the width of a human hair, yet built with perfect consistency across millions of cells. Unlike pigment-based colors that fade with time, these structural colors can persist for years, even in dried specimens, because the physical structures remain intact long after the flower's death. The discovery of these mechanisms has inspired human technologies, from color-shifting paints to more efficient solar panels, that mimic the light management strategies of petals. Some flower buds remain closed for months, waiting for one specific signal to open. Certain desert species time their blooming to rainfall, remaining dormant as tight buds until water arrives. The buds can persist in this waiting state for extraordinarily long periods, sometimes years, their tissues barely alive, metabolism slowed to a crawl. Then, when rain finally soaks the soil, chemical changes cascade through the plant, triggering rapid growth. Within days, sometimes hours, the buds swell and burst open, transforming barren landscapes into temporary gardens. The synchronization across thousands of individual plants creates floral displays visible from space. Carpets of color spreading across desert basins that were brown and lifeless the week before. The flowers rush through their entire reproductive cycle, blooming, pollination, seed production, in a matter of weeks, racing against the inevitable return of drought. This strategy requires the buds to sense and remember environmental conditions, integrating information about moisture, temperature, and day length to make high-stakes decisions about when to commit their precious resources to reproduction. The patience encoded in these waiting buds is remarkable, a botanical form of hope that persists through seemingly endless dry spells. Certain flowers generate heat that melts snow around their stems. In alpine meadows and Arctic tundra, some species warm themselves through metabolic processes, usually associated with animals. Skunk cabbage produces enough heat to melt through frozen ground and overlying snow, creating small clearings where the flower emerges weeks before surrounding plants. The warmth comes from rapidly metabolizing stored starches, essentially burning sugar to generate heat, just as our bodies do. Temperatures inside these flowers can exceed the surrounding air by 30 degrees or more, creating tropical microclimates in freezing environments. The heat serves multiple purposes. It protects delicate reproductive tissues from frost damage, volatilizes scent molecules to attract the few pollinators active in cold conditions, and provides warm landing platforms where cold-blooded insects can raise their body temperatures enough to fly. Bees visiting these flowers on frigid mornings warm themselves like travelers at a campfire before continuing their pollen gathering rounds. The energy cost of this heating is enormous, requiring the plant to divert resources that could otherwise support growth or seed production. But in environments where the growing season is measured in weeks, being first to bloom can mean the difference between reproductive success and failure. Some petals guide pollinators using patterns visible only in ultraviolet light. What appears to human eyes as a uniform yellow flower reveals itself to a bee as a complex target, with UV-absorbing zones, creating contrasting runways that point toward nectar rewards. These invisible patterns, called nectar guides, function like airport landing lights, directing visitors to the precise location where they'll contact pollen-bearing structures. The guides can be lines, dots, or concentric rings, each pattern optimized for particular pollinator types. Flowers visited by long-tongued bees often display different guides than those serving short-tongued species, reflecting the different approach angles and body positions of their respective partners. The UV patterns remain invisible to most herbivores, allowing flowers to advertise to mutualists, while remaining cryptic to those who would damage them. Some flowers change their UV patterns after pollination, shifting from bright advertisements to dull signals that redirect pollinators toward unpollinated neighbors, improving the overall reproductive success of the population. The existence of this invisible world of floral signals, completely hidden from human perception, reminds us how much of nature's complexity operates outside our sensory awareness. What we see is only a fraction of what exists. Certain flowers track the sun's movement across the sky throughout the day. Young sunflowers demonstrate this behavior most famously, their heavy heads swiveling from east at dawn to west at sunset, following the solar arc with impressive precision. The movement, called heliotropism, arises from differential growth rates on opposite sides of the stem. Cells on the shaded side elongate faster than those on the sunny side, creating a bend that turns the flower toward light. At night, the process reverses, returning the flower to its eastward-facing dawn position. This daily cycle continues as long as the plant remains in its rapid growth phase. But once the flower matures and begins producing seeds, the tracking behavior stops, and the flower fixes its orientation, almost always facing east. The eastern preference makes sense because morning sun helps warm the flower quickly, attracting early rising pollinators and giving them warm landing platforms when cold-blooded insects are still sluggish. Studies show that sunflowers prevented from tracking the sun receive fewer pollinator visits and produce fewer seeds, demonstrating real fitness consequences of this solar following behavior. The precision of the tracking requires integration of light signals, an internal circadian clock, and growth responses, creating a simple yet elegant system for optimizing light capture and pollinator service. Some flower scents contain dozens of different chemical compounds blended in precise proportions. Rose fragrance alone includes more than 300 distinct molecules, each contributing to the overall aroma. The proportions matter tremendously. Shift one component by a few percentage points, and the scent changes from pleasantly floral to sickeningly sweet or sharply medicinal. Plants synthesize these complex bouquets using specialized cellular factories, often concentrating scent production in specific tissues like petal bases or specialized glands. The mixtures serve as chemical advertisements and filters, attracting desired pollinators while repelling unwanted visitors. Some compounds in floral scents act as insect repellents, protecting the flower from herbivores, while other compounds in the same blend attract pollinators. Certain orchids produce scents that mimic insect pheromones so accurately that male bees attempt to mate with the flowers, achieving pollination while receiving nothing in return. The complexity of these chemical deceptions, where single flowers produce and maintain dozens of compounds in precise ratios, demonstrates sophisticated biosynthetic capabilities. Fragrance research continues to reveal new compounds and new functions, showing that what we smell as a simple flower scent is actually an intricate chemical language we're only beginning to decode. Certain petals change color as the flower ages, signaling pollination status to visitors. Flowers that start white might fade to pink, then deepen to purple over several days. These color changes advertise that the flower has already been pollinated and no longer offers rewards, directing pollinators toward fresher blooms, where their visit will accomplish more. The color shifts come from pH changes in petal cells or the breakdown of existing pigments into different colored compounds. Forget-me-nots famously change from pink buds to blue flowers as cellular pH shifts, while lantana flowers transition through yellow, orange, and red stages as they age. The multicolored effect created when flowers at different life stages coexist on one plant enhances the overall display, making the plant more visible from a distance, while still providing close-up guidance about which individual flowers to visit. Some research suggests that the presence of old, spent flowers in different colors actually increases pollinator visits to the plant as a whole, with a larger, more colorful display attracting more attention, despite some of the flowers offering no reward. This challenges the assumption that plants should drop spent flowers immediately, showing instead that retaining them for their advertisement value can be the optimal strategy. Some flowers bloom for only a single day, opening at dawn and dying by nightfall. These ephemeral beauties compress their entire adult existence into hours, unfurling with the first light and beginning to wither as shadows lengthen. Morning glories demonstrate this pattern in temperate gardens, their trumpet-shaped blooms fresh at breakfast, but wilted by lunch. The brevity seems wasteful until you consider the efficiency. The plant invests minimal resources in structures that must persist only long enough to achieve pollination, allowing more energy for seed production. Someday, blooming species open in synchronized waves, ensuring that fresh flowers appear daily throughout the blooming season. The short lifespan also means the flower can remain pristine and attractive for its entire display period, never showing the wear and damage that accumulates on longer-lived blooms. For the lucky observer present during the brief window of perfection, these fleeting flowers offer a lesson in impermanence and the poignant beauty of transient things. Their very temporariness makes them more precious, teaching us to appreciate the moment because it won't return. Certain flowers can close their petals in response to rain, protecting pollen from being washed away. Tulips, crocuses, and many other species demonstrate this protective behavior, their petals folding inward when moisture appears, then reopening when conditions dry. The closure mechanism operates through changes in cell turgor pressure, with cells on the inner petal surface swelling when wet, creating a bend that pulls the petal inward. The response happens quickly enough to protect the flower during sudden storms, yet slowly enough to avoid wasting energy on false alarms from brief mist or dew. Some flowers also close at night, or on cloudy days, responses that may conserve scent compounds or protect pollen during periods when pollinators are unlikely to visit anyway. The ability to open and close repeatedly requires petals that remain flexible and responsive throughout the flower's life. Unlike species whose petals open once and then remain fixed, this flexibility comes at a cost. The specialized tissues and cellular mechanisms require energy to build and maintain. But in environments with unpredictable weather, the protection of irreplaceable pollen justifies the investment. The simple elegance of this solution, using passive swelling of cells rather than active muscular movement, shows how plants accomplish complex behaviors through straightforward mechanisms. Some flower stems contain millions of microscopic channels that transport water from roots to petals. These vascular highways, called xylem, function like the plant's circulatory system, though they operate purely through physics rather than pumping hearts. Water molecules cling to each other and to the channel walls, creating continuous columns of liquid that can stretch from deep in the soil all the way to the tip of the tallest flower. The evaporation of water from petal surfaces creates negative pressure that pulls the entire column upward, like sucking through a straw extending dozens of feet into the ground. The system is remarkably efficient, but also vulnerable. A single air bubble breaking the water column can disable an entire channel. Plants compensate by building redundant systems with thousands or millions of parallel channels, ensuring that even if some fail, others continue functioning. The narrowness of the channels is crucial. They're typically measured in micrometers, far too small to see without magnification. This tiny diameter allows surface tension and molecular cohesion to work effectively, creating the capillary action that defies gravity. When you place cut flowers in a vase, you are observing this system still functioning. The severed stem continuing to pull water upward to sustain petals for days or even weeks after the flower has been separated from its roots. Certain flowers produce nectar that varies in sweetness depending on recent pollinator visits. Some species can sense how recently they've been visited and adjust nectar production. Accordingly, concentrating sweetness in long-neglected flowers while reducing output in recently visited ones. This dynamic adjustment optimizes the plant's energy investment, avoiding waste on flowers that are already successfully attracting pollinators, while boosting the appeal of those being ignored. The sensing mechanism may involve detection of pollinator saliva or monitoring of pollen removal, with the flower essentially tracking its own pollination status. The system creates frequency-dependent selection where pollinators learn to preferentially visit the most rewarding flowers, which happen to be those most in need of service. The complexity of this feedback system, where plants monitor pollinator behavior and adjust rewards in real time, demonstrates sophisticated environmental sensing. It also creates potential for conflicts of interest, where the plant's optimal strategy differs from the pollinator's preferences, leading to evolutionary arms races, as each party tries to manipulate the other. The resulting balance represents a negotiated truce rather than perfect cooperation, with neither partner fully controlling the interaction. Some petals contain cells that focus light onto reproductive structures, like living lenses. These specialized epidermal cells have curved surfaces that concentrate sunlight onto stamens and carpels, warming them and making them more visible to approaching pollinators. The effect creates bright spots within the flower that attract attention and provide thermal rewards for cold, Jan-blooded visitors. The lens gen-like cells require precise shaping during development, with the curvature determined by controlled cell expansion and wall formation. Too flat, and they fail to focus light. Too curved, and the focal point falls in the wrong location. The precision required suggests strong selective pressure favoring flowers that get this detail right. In some species, the focused light creates temperature differences of several degrees between the focal zone and surrounding tissues, enough to significantly affect insect behavior and metabolism. Bees basking in these warm spots heat up faster, allowing them to become active earlier in cool mornings. The flowers benefit from being among the first visited when pollinators emerge, improving their chances of outcrossing with distant plants rather than nearby neighbors. This optical engineering at the cellular level demonstrates that flowers are not passive decorations, but sophisticated machines designed through millions of years of evolutionary refinement. Certain flowers release pollen only when vibrated at specific frequencies. Tomatoes, blueberries, and many other species with tubular anthers require buzz pollination, where bees grasp the flower and vibrate their flight muscles rapidly, shaking pollen loose like salt from a shaker. The pollen emerges through tiny pores at the anther tip, dusting the vibrating bee in a golden cloud. The frequency matters. Too slow and pollen remains trapped. Too fast, and it disperses wastefully into the air rather than onto the bee. Bumblebees have mastered the technique, able to tune their vibrations to match the resonant frequency of different flower types. Honeybees, surprisingly, never evolved this ability and cannot effectively pollinate buzz, gen pollination flowers, despite being accomplished at other types. The specificity of this system creates strong partnerships between particular bee species and particular plant groups, with each depending on the other's precise capabilities. For the plant, buzz pollination offers a way to ration pollen, releasing it only to visitors large and strong enough to provide good pollination service while excluding weaker or less reliable insects. The vibration requirement essentially creates a strength test that filters out freeloaders. This mechanical lock gen and gen key system operates through physics rather than chemistry, yet achieves the same result, ensuring that the flower's valuable pollen reaches the right partners. Some flowers exist in relationships so specialized that plant and pollinator cannot survive without each other. Yucca plants and yucca moths demonstrate this mutual dependency perfectly. The moth collects pollen from one flower, deliberately packs it into a ball, flies to another yucca flower, lays eggs in the flower's ovary, then carefully deposits the pollen ball on the stigma, ensuring fertilization. The developing moth larvae feed on some of the resulting seeds, but the plant produces enough extras that both partners benefit. Neither species can reproduce without the other. The yucca cannot self-gen pollinate, and no other insect performs the necessary pollen transfer, while the moth larvae can develop only inside yucca seed pods. This obligate mutualism has persisted for millions of years. cogen evolving in such precise synchronization that their life cycles align perfectly, the moths emerging exactly when yucca flowers open. The relationship is so specific that different yucca species have different moth species, each pair locked in evolutionary partnership. The system is beautiful but fragile. Disruption to either partner threatens both. Such extreme specialization represents an evolutionary gamble, trading flexibility for perfect optimization of a single strategy. In stable environments, these partnerships thrive, but rapid environmental change can sever the connection, potentially dooming both species. Certain petals repel water so effectively that raindrops bounce off without wetting the surface. This superhydrophobic property comes from microscopic bumps and waxy coatings that prevent water from making direct contact with the petal. Instead, drops sit atop the nanostructures like balls balanced on a bed of nails, maintaining spherical shapes that roll away at the slightest tilt. The water-jet repelling surface keeps petals clean, as rolling drops pick up dirt and pollen, carrying contaminants away. This self-jet cleaning property, observed in lotus leaves and many flower petals, has inspired human technologies from stain-jet-resistant fabrics to non-jet stick coatings. The combination of structure and chemistry required for superhydrophobicity shows sophisticated materials engineering at the microscopic scale. Plants cannot simply make flat surfaces and apply waterproofing. The geometry of the bumps, their spacing, height, and the chemical properties of the wax coating must all work together perfectly. Get any component wrong and water penetrates, defeating the system. The fact that countless plant species independently evolved similar solutions suggests strong selective pressure favoring water repellency, likely because wet petals attract harmful fungi and bacteria, suffer damage from freezing, and become less attractive to pollinators. Some flowers regulate their temperature as precisely as warm, jet-blooded animals. Sacred lotus maintains its blooms between 30 and 30 degrees Celsius regardless of ambient temperature, warming them when nights turn cold and cooling them during scorching days. The temperature control comes from variable metabolic rates and the evaporative cooling from water loss, creating a system that responds to thermal stress in both directions. The consistent warmth attracts beetles that serve as pollinators, providing them with comfortable environments and rewarding their visits with reliable thermal havens. The energy cost of this thermoregulation is substantial, requiring continuous burning of stored reserves throughout the flower's multi-day lifespan. The metabolic intensity rivals that of flying hummingbirds, making these flowers among the most energetically expensive structures in the plant kingdom. The precision of the regulation suggests sophisticated sensing and response systems that monitor temperature and adjust metabolism accordingly. The mechanisms remain incompletely understood, but clearly involve coordination across multiple cellular systems. The parallel between plant thermoregulation and animal endogamy shows that similar environmental challenges can drive convergent evolution of similar solutions, even in organisms as different as lotus and mammals. Certain flowers produce electrical fields that bees can detect and interpret. The flowers carry slight negative charges, while bees flying through the air accumulate positive charges, creating measurable electrical gradients between approaching pollinators and awaiting blooms. When a bee lands, the electrical field changes momentarily, and this change can persist for up to 2 minutes. Other bees can sense these altered fields and use the information to avoid recently visited flowers that are likely depleted of nectar. The system provides a faster gen updating signal than scent marks, giving bees real gen-time information about patch quality. The discovery of these electrical signals open new understanding of the sensory world of pollinators, revealing communication channels humans cannot directly perceive. The implications extend beyond simple efficiency. Electrical fields might convey information about flower species, individual identity, or even flower health. The field is literally an extension of the flower into surrounding space, creating a zone of influence that begins affecting pollinator behavior before physical contact occurs. This electrical dimension of flowers adds another layer to the already complex signals plants use to attract, guide, and inform their animal partners. Some flower buds protect themselves with sticky resins that trap small predators. The gummy coating on poppy buds and many other species serves as both chemical defense and physical barrier, deterring chewing insects while sealing the developing flower against fungal invasion. Tiny ants and beetles that attempt to walk across these surfaces become hopelessly mired, dying stuck to the very structure they sought to attack. The resin production requires specialized glands that synthesize and secrete complex mixtures of sticky compounds, an energy investment justified by the protection it provides to irreplaceable reproductive tissues. Some resins contain antimicrobial chemicals that actively kill bacteria and fungi, combining physical barriers with chemical warfare. The stickiness decreases as the flower matures and opens, suggesting controlled degradation of the defensive coating. Once the vulnerable bud stage passes, bees and other legitimate pollinators visiting mature flowers encounter minimal stickiness, allowing them to move freely, while the flower was protected during its most vulnerable early development. The timing of this protective system shows sophisticated developmental programming, where defensive investments peak when threats are greatest and diminish when the costs outweigh benefits. Certain flowers can orient their petals independently, creating complex, three-dimensional forms, rather than opening symmetrically like simple bowls. Some species create intricate shapes with petals angled in different directions, each positioned to serve specific functions. Orchids demonstrate this most dramatically, with specialized petals forming landing platforms, nectar guides, and even trap-jam-like structures that temporarily confine pollinators. The asymmetry creates directionality, guiding visitors into the flower from one specific angle and directing them out along a path that ensures contact with pollen. The positioning of each petal is precisely controlled during development, with genetic programs specifying not just size and color, but also three-dimensional orientation. The resulting architectures rival human engineering in their complexity and precision. Some orchid flowers contain moving parts that deposit pollen packets onto pollinators, with spring-loaded mechanisms of remarkable sophistication. The specialization can become so extreme that individual flowers function only with single pollinator species, creating partnerships of extraordinary specificity. The complexity demonstrates that flowers are not simple, static structures, but dynamic, precisely engineered devices designed to accomplish specific tasks with maximum efficiency. Some petals fluoresce under ultraviolet light, creating patterns invisible in normal daylight. Certain molecules in petal cells absorb UV radiation and emit it as visible light, making the flowers glow with eerie beauty when illuminated by black lights. The fluorescence may enhance visibility to pollinators with UV-sensitive vision, creating super-Zen stimuli that attract more visits than non-Zen fluorescent competitors. The patterns revealed by fluorescence sometimes differ from visible Zen light patterns, adding additional layers of information to the flower's advertisement. Some species show fluorescent nectar guides that lead pollinators inward with glowing paths. The discovery of widespread fluorescence in flowers is relatively recent, suggesting that we still have much to learn about how plants communicate visually. The phenomenon connects to the structural colors mentioned earlier, showing that flowers manipulate light in multiple ways simultaneously, reflecting, absorbing, transmitting, diffracting, and fluorescing, to create the visual displays we perceive. Each of these optical effects requires specific cellular architectures and chemical compositions, representing evolutionary investments in increasingly sophisticated signaling systems. Certain flowers respond to carbon dioxide exhaled by approaching pollinators. Some orchids can detect the CR in a bee's breath and respond by altering scent production or triggering mechanical movements. The sensitivity to such subtle cues suggests that flowers actively monitor their surroundings for potential visitors, rather than simply displaying static advertisements. The detection mechanism likely involves the same see-all sensors that help the plant regulate photosynthesis. Repurposed for detecting animals, this dual use of sensory systems shows evolutionary efficiency, where existing capabilities are adapted for new functions, rather than building entirely new systems from scratch. The response to pollinator presence allows the flower to time its displays precisely, perhaps intensifying scent or opening structures only when visitors are actually present, conserving resources during periods when no pollinators are near. This conditional signaling represents a more sophisticated strategy than constant display, showing that plants possess capabilities we're only beginning to appreciate. Some flowers produce oils instead of nectar, attracting specialized bees that collect oils for larval provisions. The oils are thick, lipigen-rich substances quite different from sugary nectar, requiring pollinators with specialized collection equipment. Oil-gen collecting bees have evolved specially modified leg hairs that soak up and transport these viscous rewards. The flowers producing oils often have distinctive morphologies with specific landing platforms and oil-gen secreting glands positioned to contact the bee's collection apparatus. The oil-gen for-gen pollination trade represents an alternative economy running parallel to the more common nectar-gen for-gen pollination system. Neither plant nor bee can participate in both systems simultaneously. The flower produces either oil or nectar, and bees are equipped to collect one or the other, but not both. This specialization creates distinct communities of oil flowers and oil bees, groups that rarely interact with the nectar-gen-based system. The existence of these parallel economies shows how diversification in rewards can reduce competition and allow more species to coexist, each serving different market niches. Certain petals contain hairs that trap heat and create warm microclimates. Fuzzy flowers, like many alpines, sport dense coats of fine hairs that insulate delicate reproductive parts, allowing them to function in environments where temperatures regularly drop below freezing. The hairs trap still air next to the flower's surface, creating an insulating blanket that can keep flower interiors several degrees warmer than surrounding conditions. This simple structural adaptation allows flowers to bloom weeks earlier than naked species, taking advantage of briefly favorable conditions in otherwise harsh environments. The hairs may also protect against excessive UV radiation at high altitudes where atmospheric shielding is minimal. Some visitors seek out these hairy flowers specifically for the warmth they provide, creating partnerships where the plant offers thermal rewards in exchange for pollination service. The hairiness comes in countless variations, short or long, sparse or dense, straight or curled, each variant representing a different optimization for particular environmental conditions. The diversity shows how even simple structures like hairs can be elaborated into sophisticated adaptations through evolutionary tinkering. Some flowers change their displays based on whether they contain pollen or nectar rewards. Flowers that have already released their pollen, but still produce nectar, might shift their color or scent to signal the change in available rewards. This honest advertising helps pollinators make informed decisions about which flowers to visit based on their current needs. Pollen collectors avoid nectar-gen-only flowers, while nectar feeders skip pollen-gen-only blooms, improving efficiency for both parties. The ability to signal reward status requires the plant to monitor its own condition and alter displays accordingly, showing a form of self-gen awareness at the flower level. The honesty of these signals is maintained by the cost of deception. Flowers that falsely advertise rewards quickly lose pollinator interest as visitors learn to avoid them. This creates pressure for truthful signaling, where flowers accurately report their current offerings. The system demonstrates how communication can evolve even between parties with somewhat conflicting interests, with honesty maintained through the mutual benefits of accurate information exchange. Certain flowers bloom in synchronized waves across entire populations. All individuals of a species in a region might flower simultaneously for just a few days, then remain dormant for the rest of the year. This mass flowering overwhelms, seed predators with superabundant resources, ensuring that enough seeds escape consumption to regenerate the population. The synchronization requires environmental cueing that all individuals can detect, such as temperature thresholds, day length, or rainfall patterns. Some tropical trees bloom synchronously in response to drought stress or sudden temperature drops, with entire forests bursting into flower within days of the triggering event. The coordination creates spectacular displays and intense competition for pollinators, with individual flowers striving to stand out in the crowd. The strategy works best in stable environments where the cueing events occur predictably, allowing plants to anticipate and prepare for the brief flowering window. Mass flowering represents a bedtime hedging strategy, where individual plants sacrifice the advantages of unique timing in exchange for the protection and pollinator saturation benefits of synchrony. Some petals serve as solar panels, capturing light energy that warms the flower's center. The curved shapes and reflective surfaces of certain flowers focus sunlight onto reproductive structures, raising their temperature and accelerating pollen maturation. Arctic poppies track the sun just like sunflowers, their bold gen-shaped flowers acting as parabolic reflectors that concentrate light into a bright focal point, where temperatures can exceed ambient by 10 degrees Celsius or more. Insects visiting these warm spots heat up rapidly, enabling flight in conditions that would otherwise keep them grounded. The flowers essentially offer heating services in exchange for pollination, creating partnerships based on thermal rewards rather than food. The solar concentration requires precise petal shaping and positioning, with the optimal geometry changing as the flower matures and the position of reproductive structures shifts. Some flowers track the sun with their heat-focusing geometry early in the day, but then fix their orientation once pollinators become active, suggesting complex prioritization of different functions across the day. Certain flowers produce sounds at frequencies below human hearing. These infrasonic vibrations, typically created by wind, moving through complex flower structures, may serve as long-distance communication signals, or simply be unavoidable byproducts of morphology. Some evidence suggests that root systems might detect these vibrations and use them to locate nearby plants, though the mechanisms and functions remain speculative. The possibility that flowers communicate through sounds we cannot hear mirrors the discoveries of UV patterns and electrical fields, repeated revelations that plants operate in sensory realms invisible to us. Each new discovery of hidden communication channels reshapes our understanding of plant sophistication, forcing us to abandon simplistic views of flowers as passive, mechanical structures. The more we learn about the sensory worlds of plants, the more apparent it becomes that they actively monitor and respond to their environments in ways rivaling the complexity of many animals. Some flowers practice precise timing, opening and closing at specific hours, so reliably that you could set clocks by them. Morning glories, as their name suggests, open with dawn light and close by afternoon. Other species open only in afternoon, evening, or midnight, each timing its display to match the activity patterns of its specific pollinators. Carolus Linnaeus, the father of botanical classification, designed a flower clock with species arranged in a circle, each blooming at a different hour, the progression marking time's passage through the day. The precision of these rhythms comes from internal circadian clocks that continue running even when plants are isolated from external time cues. The molecular mechanisms involve feedback loops of gene expression that cycle with roughly 24-hour periods, self-sustaining oscillators that synchronize to environmental light-dark cycles, but persist without them. The fact that flowers possess these sophisticated timekeeping systems shows the importance of precise temporal coordination with pollinator activity and optimal environmental conditions. Certain petals contain crystals that make them sparkle in sunlight. These microscopic mineral formations, often calcium oxalate, catch and reflect light in glittering displays. The sparkle might attract pollinators or simply be an unavoidable byproduct of calcium metabolism. But it adds another dimension to floral visual displays. The crystals form in specific patterns within cells, sometimes as needles, sometimes as clusters, each arrangement creating different optical effects. Some sparkle primarily at specific angles, creating flowers that flash as viewers move past them. The dynamic quality of sparkling flowers makes them particularly attention-grabbing, with the movement-dependent shimmer potentially triggering stronger responses in pollinator visual systems than static colors would. The crystalline structures also provide mechanical strength to cells and may serve defensive functions by making tissues unpalatable to herbivores. As with many floral features, sparkle likely serves multiple simultaneous functions, with the balance of costs and benefits determining how elaborate these crystalline displays become. Some flowers can count, keeping track of how many times they've been visited and responding differently after specific numbers. Certain orchids release less scent after receiving multiple visits, apparently detecting through pollen removal or other cues that they've achieved sufficient pollination. The counting mechanism remains mysterious but clearly involves some form of cellular memory that accumulates across individual pollinator visits. Other flowers increase nectar production after visits, essentially rewarding pollinators who return repeatedly. The different strategies reflect different pollination biologies. Some species need just 1 or 2 visits to achieve full seed set, while others benefit from many visits over extended periods. The ability to count visits and modulate responses accordingly shows numerical cognition in plants, a capability once thought limited to organisms with nervous systems. The discovery challenges assumptions about the cognitive requirements for counting and suggests that relatively simple biochemical systems can track and respond to quantitative information. Certain flowers create illusions, visually mimicking other species or even other kingdoms of life. Some orchids so perfectly resemble female insects that males attempt to mate with them, achieving pollination, while the flower provides no reward whatsoever. The deception requires extraordinary precision in shape, color, texture, and even scent, with the orchid producing chemical compounds identical to insect pheromones. The accuracy of these mimicries suggests intense selective pressure from discriminating pollinators, who abandon flowers that don't quite match their expectations. The evolutionary arms race between deceptive flowers and increasingly choosy pollinators drives continuous refinement of the mimicry, producing flowers that fool even expert human observers. Other flowers mimic dung or rotting meat, attracting flies as pollinators with false promises of egg-laying sites. These carrion flowers produce not just visual mimicry, but also odors of decay so convincing that flies lay eggs on them, condemning their larvae to starvation when the deception is revealed. The fact that such complex multisensory deceptions evolve repeatedly across plant lineages shows that exploitation of pollinator preferences can be a successful strategy, despite its dishonesty. Some petals function as landing guides, their shapes and patterns literally directing pollinators to proper approach paths. Snapdragons create landing platforms that open under a bee's weight, allowing access to nectar while simultaneously dusting the insect with pollen. The mechanical coordination required for this system is remarkable. The landing platform must be strong enough to support the visitor yet flexible enough to open, and the geometry must align pollinator body and pollen-bearing structures precisely. Different-sized bees fit the mechanism differently, with some too small to trigger the opening and others too large to fit through the entrance. This size selectivity ensures that only appropriate pollinators can access rewards and accomplish pollination. The flower essentially creates a physical lock that only specifically shaped keys can open, a widespread strategy seen in countless species. The elaboration of flower shape into complex, functional mechanisms challenges the view of petals as mere colorful decorations, revealing them as precisely engineered tools. Certain flowers communicate with their neighbors chemically, warning of herbivore attacks. When damaged by caterpillars or other chewing insects, some flowers release volatile compounds that trigger defensive responses in nearby plants. The warned neighbors increase production of toxic or distasteful chemicals, making themselves less palatable before the herbivores arrive. The system works because herbivores rarely stop at one flower, so neighbors of attacked plants are likely to be visited soon. By preparing defenses preemptively, the warned plants reduce damage and improve survival. The communication is eavesdropping rather than true cooperation. The damaged plant releases volatiles as a side effect of its own defensive responses, and neighbors exploit these inadvertent signals. Nonetheless, the result is a coordinated defensive response across groups of plants, creating neighborhoods of heightened resistance that herbivores find difficult to penetrate. The discovery of this plant communication revolutionized understanding of plant interactions, showing that they're not isolated individuals, but rather members of complex communities that share information. Some flowers protect themselves with decoy structures that draw herbivores away from essential organs. Extra petals, false stamens, or other expendable tissues absorb damage that would otherwise affect reproduction. The decoys might be positioned at the flower's periphery, where approaching herbivores encounter them first, or might actively attract attention through bright colors or enhanced scent. Sacrificing these structures allows the flower to continue functioning reproductively, despite ongoing herbivore pressure. The strategy makes sense when producing replacement decoys cost less than the reproductive loss from damage to core structures. Some flowers continuously produce new decoy petals throughout their blooming period, maintaining protection despite steady attrition. The presence of these specialized defensive structures shows that flowers face constant threats and have evolved diverse strategies beyond chemical toxins for managing herbivore damage. Certain petals contain antifreeze proteins that prevent ice crystal formation in sub-zero temperatures. These specialized molecules disrupt the normal crystallization of water, keeping cellular fluids liquid well below their normal freezing point. The protection allows flowers to survive and function during frost events that would kill unprotected tissues. The antifreeze proteins are similar to those found in cold water fish and some insects. Examples of convergent evolution, finding the same molecular solution to freezing stress across vastly different organisms. The proteins must be continuously synthesized and maintained, representing an energetic cost. But in environments with frequent frost, this investment is clearly worthwhile. Some flowers combine antifreeze proteins with the thermal generation discussed earlier, using both passive chemical protection and active heating to survive extreme cold. The multi-layered defenses demonstrate the importance of cold tolerance for early blooming species. Some flowers produce rewards specifically for ants, which then defend the plant against larger herbivores. These extrafloral nectaries, located outside the actual flower on stems or leaves, provide sugar to ant colonies that patrol the plant aggressively. The ants attack any caterpillar or beetle they encounter, biting and spraying acid to drive away threats. The plant essentially hires a security force, paying in sugar for protection services. The partnership benefits both parties. The plant suffers less herbivore damage, while the ants gain a reliable food source. The specificity of these arrangements varies, with some plants attracting any ant species while others partner with particular types that provide the most effective defense. The most sophisticated mutualisms involve plants that provide not just food but also housing, growing hollow thorns or stems where ant colonies nest. These ant plants achieve remarkable protection, but become dependent on their partners, sometimes unable to survive without them. The partnerships can last for the life of both organisms, creating long-term alliances that shape both partners' evolution. Certain flowers guide pollinators using temperature gradients, creating warm zones that lead to nectar. Pollinators with heat-sensitive organs can detect these thermal patterns and follow them like thermal breadcrumb trails. The warmth might come from focused sunlight, metabolic heat generation, or dark pigments that absorb solar radiation. Whatever the source, the result is a flower that broadcasts not just visual and chemical signals, but thermal ones as well. The multimodal signaling may be redundant, with different signals conveying the same information, or might communicate different messages to different pollinator types. Bees with good color vision might rely primarily on visual cues, while beetles with poor vision but good heat sensing follow thermal signals to the same destination. By providing multiple signal types, flowers can appeal to diverse pollinator communities, improving their chances of attracting visits, even as pollinator composition shifts seasonally or across years. Some petals change thickness during the day, swelling at dawn and thinning at dusk. The changes come from water movement into and out of petal cells, controlled by osmotic pressures that vary with light and temperature. The thickening might improve the petal's structural integrity during peak pollinator activity, or enhance its visual appearance through changes in light transmission. The daily cycle of swelling and shrinking continues throughout the flower's life, responding to environmental conditions rather than following a fixed program. During drought, petals might remain thinner as the plant conserves water, while abundant moisture allows maximum swelling. The flexibility allows the flower to balance competing demands of display, water conservation, and structural integrity, optimizing the trade-offs in real time based on current conditions. Certain flowers produce different colored petals from identical genetic information based solely on soil chemistry. Hydrangeas famously shift from pink in alkaline soil to blue in acidic conditions, the same plant producing different hues depending on environmental pH. The color change comes from anthocyanin pigments interacting with aluminum ions that become available in acidic conditions. The identical genes create different outcomes depending on the chemical environment, an example of phenotypic plasticity where a single genotype produces multiple phenotypes. The adaptive value of this flexibility is unclear. It might simply be an unavoidable consequence of the chemistry involved rather than serving specific functions. Nonetheless, the phenomenon demonstrates that flower color isn't always determined solely by genetics, but can be significantly influenced by environmental factors. This means that the same flower could look completely different if transplanted to different soil, a reminder that organisms are products of both their genes and their environments. Some flowers possess memory systems that allow them to remember stressful events and prepare for their recurrence. Plants that experience drought during bud formation sometimes produce flowers with thicker cuticles or altered stomatal densities, modifications that persist even after water becomes abundant again. The changes suggest the flower remembers the stress and adjusts its construction to better handle similar challenges in the future. The memory operates through epigenetic modifications, chemical tags attached to DNA that alter gene expression without changing the underlying genetic sequence. These modifications can sometimes persist across cell divisions, maintaining the stress memory as the flower grows. Some evidence suggests that stress memories might even transfer to seeds, potentially allowing parent plants to prepare offspring for environmental conditions they're likely to encounter. This transgenerational preparation would represent a form of anticipatory parenting, where adults provide offspring not just with genetic information and stored nutrients, but also with environmental predictions that improve offspring survival. Certain petals exhibit iridescence that shifts through the spectrum as viewing angle changes. These rainbow effects come from multiple layers of cells with different refractive indices, creating thin film interference, similar to oil slicks or soap bubbles. The precise spacing of these layers determines which wavelengths undergo constructive or destructive interference, producing colors that exist nowhere in the petal's chemical composition. The iridescence is purely structural, arising from physics rather than pigments. As you move around these flowers, the colors shift: blue becomes green, becomes gold, creating dynamic displays that attract attention far more effectively than static hues. The engineering required to build multi-layered cells with precise spacing across millions of petal cells demonstrates remarkable developmental control. The structures must form correctly during the brief period of petal growth, with no opportunity for adjustment once construction completes. The reliability of this self-assembly process, achieving consistent results across countless flowers, shows the robustness of the genetic programs controlling cellular architecture. Some flowers redirect wind flow through their structures, creating calm zones where pollinators can land safely. Complex petal arrangements and protective bracts shield central flower parts from strong winds that would make landing difficult or impossible. Bees approaching these flowers find unexpected stillness within the protected zone, allowing precise positioning despite turbulent conditions just inches away. The wind-baffling architecture requires careful three-dimensional design, with structures positioned to deflect airflow around rather than through the flower center. Different wind directions require different protective geometries, so flowers in exposed locations often develop asymmetric forms that preferentially protect against prevailing winds. The trade-off is that some approach directions remain exposed and difficult, potentially biasing pollinator traffic patterns. Plants might solve this by orienting flowers to present protected approaches toward the direction from which pollinators typically arrive, integrating multiple environmental factors into optimal flower positioning. Certain flowers can detect gravity and orient their blooms accordingly. Downward-facing flowers use gravitropic sensors to maintain their nodding posture as stems grow and reposition the flower. The sensors involve specialized cells containing starch grains that settle under gravity's influence, their position triggering differential growth that rights the flower or maintains its desired orientation. The same sensory systems that help roots grow down and shoots grow up are repurposed in flowers to control display angle. Some species prefer upward-facing blooms that catch rain and display their interiors, while others hang downward to protect pollen from moisture and present their undersides. The optimal orientation depends on pollinator preferences and environmental challenges, with evolution fine-tuning gravitropic responses to achieve species-specific display angles. The precision of these systems allows flowers to maintain characteristic orientations, despite being jostled by wind, visited by heavy pollinators, or growing on sloping terrain that would passively tilt them in non-adaptive directions. Some petals function as drums, their thin surfaces amplifying the sounds of visiting pollinators. The vibrations created by bee wing beats and movements resonate through stretched petal membranes, creating sounds louder than the insect alone would produce. Other bees may hear these amplified sounds from greater distances, potentially using them to locate productive foraging areas. The flowers essentially become acoustic beacons, broadcasting the presence of active pollinators to other potential visitors. Whether this acoustic advertising benefits the plant, the pollinators, or both remains unclear. Plants might benefit from increased visitation rates, while bees gain information about competitor presence and resource availability. The unintentional sound production could also be neutral or even costly, an unavoidable consequence of building thin, flexible petals that happen to vibrate when disturbed. Determining the adaptive significance of these acoustic properties requires careful experimentation. But regardless of function, the phenomenon adds another dimension to the sensory complexity of flowers. Certain flowers produce latex or sticky saps that trap small insects. Though unlike true carnivorous plants, they don't digest them. The trapped insects may deter other herbivores through their gruesome display, warning potential attackers of the flower's defensive capabilities. Alternatively, the trapping might be incidental, an unavoidable side effect of producing sticky defensive compounds that happen to immobilize small insects. The distinction between defensive trapping and carnivory becomes blurred in some species that show intermediate traits, trapping insects and absorbing some nutrients from their decomposition, but lacking the specialized digestive systems of true carnivores. These boundary cases remind us that evolutionary strategies exist along continuums rather than in discrete categories. What begins as simple defensive secretions might gradually evolve toward carnivory if mutations arise that allow nutrient absorption from trapped prey. The prevalence of sticky insect-trapping flowers that aren't carnivorous suggests that the full transition to carnivory requires overcoming significant barriers, with most lineages never completing the journey. Some flowers produce patterns visible only when wet, revealing hidden advertisements during rain. The moisture changes the optical properties of petal surfaces, making normally invisible markings become visible through altered reflectance or transparency. These rain-activated patterns might guide pollinators that are active during wet conditions, or might simply be non-functional byproducts of petal architecture. The appearance of patterns when wet suggests that petal surface structures have different optical properties than underlying tissues, with water filling microscopic spaces and changing how light interacts with these layered materials. The complexity of petal architecture continues to surprise researchers, with each new analytical technique revealing previously hidden structures and patterns. What appears simple to the naked eye becomes intricate when viewed under microscopes or in different wavelengths, showing that flower petals contain far more information than casual observation reveals. Certain flowers adjust their nectar chemistry based on ambient temperature. During cool periods, they produce more concentrated sugars that provide higher energy rewards, compensating pollinators for the additional metabolic costs of flying in cold conditions. When temperatures rise, the nectar becomes more dilute. Matching pollinator needs to hydration as well as energy. This dynamic adjustment requires monitoring temperature and altering the chemistry of secreted nectar accordingly, showing sophisticated environmental sensing and response. The benefits accrue primarily to pollinators, which receive optimally formulated rewards across varying conditions. But plants also benefit indirectly by maintaining consistent pollinator service despite temperature fluctuations. The stability of pollination service improves plant reproductive success, justifying the costs of dynamic nectar adjustment. This represents another example of plants providing variable rewards that match current conditions, rather than fixed offerings that might be suboptimal under changing circumstances. Some petals contain specialized cells that produce volatile compounds only when crushed or damaged. These break and release mechanisms ensure that defensive chemicals are deployed exactly when and where they're needed, at the site of herbivore attack. Undamaged portions of the flower avoid the costs of continuous chemical production and potential self-toxicity. When a caterpillar chews through these cells, they rupture and release concentrated toxins or deterrents directly into the herbivore's mouth. The immediate negative feedback may cause the herbivore to cease feeding and move elsewhere, limiting damage to the attacked flower. Some of these break and release compounds also serve as volatile signals that attract predatory insects that hunt herbivores, recruiting additional defenses from the broader ecosystem. The multifunctionality of these chemical systems shows the economy of evolution, where single compounds serve multiple roles rather than requiring separate systems for each function. Certain flowers bloom in sequence across a single plant, opening a few at a time rather than all at once. This staggered blooming extends the flowering period, increasing the chances of encountering pollinators during at least some of the bloom time. It also spreads the resource investment across time, avoiding the need to support many open flowers simultaneously. For pollinators, sequential blooming provides reliable resources over extended periods, encouraging repeated visits to the same plant. The predictability benefits both parties. Plants receive consistent pollination service, while pollinators gain dependable food sources. The genetic programming required to create this sequential pattern involves complex developmental controls that specify which buds open, when often in specific spatial patterns, like bottom to top or center to periphery. The precision of these programs shows that flowering phenology is not random but highly orchestrated to optimize reproductive success. Some flowers create microclimates within their structures that differ dramatically from surrounding conditions. Deep tubular flowers, or enclosed blooms, maintain temperature and humidity levels distinct from external air, creating pockets of favorable conditions. Pollinators entering these spaces experience sudden environmental shifts: cooler, more humid air, or warmer, drier conditions depending on flower architecture and metabolism. The microclimates may make visits more comfortable for pollinators or might serve the flower's needs for protecting pollen or developing seeds. Either way, the ability of flowers to create and maintain distinct internal environments shows that they're not simply passive structures at equilibrium with surroundings, but active modifiers of local conditions. The energy costs of maintaining these environmental differences must be substantial, suggesting that the benefits, whether through pollinator attraction or reproductive structure protection, are correspondingly large. Certain petals exhibit dichroism, displaying different colors when viewed in different lighting conditions. Under direct sunlight, they might appear one color, while diffuse shade reveals completely different hues. The effect comes from pigments, or structures that respond differently to the polarization, intensity, or spectral composition of different light sources. The adaptive significance remains unclear. It might enhance visibility across varying conditions, confuse herbivores, or simply be a neutral byproduct of petal chemistry. For human observers, dichroic flowers present changing appearances as clouds pass overhead, or as viewing position shifts them between sun and shadow, creating dynamic displays that hold attention through their unpredictability. The phenomenon reminds us that color is not an intrinsic property of objects, but rather an interaction between material properties, illumination characteristics, and observer sensory systems. Different observers with different visual systems might perceive the same dichroic flower. Some flowers produce modified petals that mimic stamens, creating false advertisements of abundant pollen. These pseudostamens attract pollen, collecting bees that discover upon landing that the apparent pollen source is actually infertile tissue. Despite the deception, some pollination may occur as the bees investigate the flower searching for real pollen. The false stamens represent investment in deception that pays off if it increases visits sufficiently to offset the costs of building non-functional structures. The evolutionary stability of this strategy depends on the frequency of deception. If too many flowers cheat, pollinators learn to avoid them, and the strategy fails. This creates frequency-dependent selection, maintaining a mix of honest and dishonest signalers in the population. A dynamic equilibrium that shifts as pollinator learning and flower traits co-evolve. Certain flowers demonstrate thigmomorphogenesis, altering their growth in response to touch. Flowers that experience frequent contact from visitors or wind grow shorter, sturdier stems with thicker tissues compared to those developing in protected locations. The mechanical stimulation triggers developmental changes that reinforce the plant against the very forces that stimulated the response. This adaptive plasticity allows individual plants to fine-tune their construction to match local conditions without requiring genetic differences. A single genotype produces different phenotypes in exposed versus protected locations, optimizing the trade-off between height for visibility and competitive advantage and sturdiness for resistance to mechanical damage. The memory of past mechanical stress continues influencing development even after the stimulation ceases, showing integration of environmental information over time rather than simple moment-to-moment responses. Some petals contain light-piping structures that channel sunlight toward the flower center. Similar to fiber optic cables, specialized cells with high refractive indices guide light internally through total internal reflection, directing photons from petal surfaces down to reproductive structures. The focused light provides energy for warming or may enhance visibility of central flower features. The evolution of such sophisticated optical engineering in plants seems remarkable until we remember that millions of years and countless trillions of individual flowers provide ample opportunity for natural selection to stumble upon and refine effective solutions. The light-piping cells require precise shaping and optical properties, but the genetic programs creating them can be surprisingly simple, with complex structures emerging from straightforward rules applied during development. The apparent sophistication of the result sometimes obscures the simplicity of the underlying mechanisms. Certain flowers can sense the weight of visitors and respond to discriminate between pollinators and nectar thieves. Flowers visited by sufficiently heavy insects might increase nectar production on subsequent days, while those experiencing only light touches from small, ineffective visitors reduce investment. The weight sensing likely operates through mechanical deformation of tissues that triggers biochemical cascades translating physical force into cellular signals. The discrimination based on visitor weight provides a rough proxy for visitor effectiveness. Lager bees generally accomplish better pollination than tiny flies, or beetles that may consume nectar without contacting reproductive structures. By rewarding effective pollinators with increased nectar and starving out ineffective ones, flowers shape their visitor communities toward optimal compositions. This represents active management of pollinator relationships rather than passive acceptance of whoever arrives. Some flowers produce heat impulses synchronized across populations, creating waves of warmth that sweep through meadows. The coordination might come from environmental cueing that synchronizes individuals, or from direct thermal signaling, where heat from one flower triggers neighbors to begin their own thermogenesis. The resulting waves create spatio-temporal patterns of temperature variation that might influence pollinator movements, or simply be epiphenomenal. The existence of synchronized heating suggests that flowers monitor thermal conditions and potentially detect heat from neighbors, adding thermal sensing to the growing list of environmental variables that flowers can perceive and respond to. The mechanisms underlying this synchronization remain largely mysterious, representing one of many aspects of flower biology, where we've observed the phenomenon but don't yet understand the underlying processes. Certain petals function as humidity sensors, changing color or conformation in response to moisture levels. The changes might help protect pollen during humid conditions, when viability is threatened, or might signal moisture status to pollinators whose own activities are moisture-dependent. The sensing mechanism could operate through hygroscopic movement of specialized tissues that swell or shrink with humidity changes, passively transducing environmental information into visible signals. No active metabolism is required. The physics of water absorption drives the response. This passive sensing shows that flowers can respond to environmental variation without continuous energetic investment, using material properties to achieve effective monitoring. The distinction between active sensing requiring metabolic energy and passive sensing using physical properties becomes blurred when considering the energetic costs of building the specialized tissues that enable passive sensing. Both strategies require investment, just at different stages, continuous operating costs versus upfront construction costs. Some flowers attract pollinators by mimicking the appearance of food sources the pollinators are already searching for. Flowers that resemble fruits might attract fruit, feeding insects, while those mimicking aphid honeydew deposits appeal to honeydew-feeding species. The mimicry exploits existing pollinator foraging preferences rather than creating new associations, allowing the flower to tap into established behavioral patterns. The effectiveness depends on the fidelity of the mimicry close. Matches receive more visits while poor imitations are ignored. This creates selection for increasingly accurate mimicry, driving evolutionary refinement toward perfection. However, perfection is never quite achieved because random mutations continuously introduce variation, while changing pollinator preferences create a moving target. The result is ongoing evolution rather than stable endpoints, with flowers and their models engaged in endless pursuit. Certain flowers produce antibiotic compounds in their nectar that reduce pathogen loads in visiting pollinators. Bees consuming this medicated nectar show lower infection rates from gut parasites and other diseases. The flowers may benefit from healthier pollinator populations that provide better service, or the antibiotics might simply protect the nectar itself from microbial spoilage. Either way, the phenomenon demonstrates that floral rewards can be more complex than simple sugars, containing bioactive compounds that influence pollinator health. The discovery has sparked interest in using flower-derived compounds as natural medicines, both for managed pollinators like honeybees and potentially for human applications. The chemical diversity of floral nectars remains incompletely explored, with new compounds and bioactivities discovered regularly as researchers apply increasingly sophisticated analytical techniques. Some petals exhibit phototropism independent of the rest of the plant, bending to track light sources. Individual petals can orient themselves toward optimal illumination, while the flower overall maintains a different position, creating complex geometries where petals twist and curl in various directions. The independent movement of petals requires localized sensing and response systems within each petal, showing that flowers possess distributed control rather than centralized command structures. Each petal essentially makes its own decisions about optimal positioning based on local conditions. The resulting flower architecture emerges from the collective action of all petals, rather than being imposed by an overall plan. This decentralized organization resembles swarm intelligence in social insects, where colony-level patterns emerge from individuals following simple local rules without any overarching coordination. Certain flowers can remain viable for years as dried specimens, then revive when given water. These resurrection flowers survive extreme dehydration by entering dormancy states where metabolism nearly ceases. Some flowers create visual illusions of depth and shadow using only flat, two-dimensional petal surfaces. Carefully graduated color patterns create false perspectives that make portions of the flower appear three-dimensional when they're actually planar. The illusions might make flowers appear larger than they actually are, or create the impression of complex structures from simple forms. The adaptive value of these visual tricks remains speculative, but they demonstrate that flowers can manipulate pollinator visual perception, essentially hacking the image processing systems of insect brains to create perceptions that don't match physical reality. The sophistication of some of these illusions rivals human artistic techniques like trompe-l'oeil, showing that evolution can discover principles of visual perception and exploit them to advantage. Certain flowers demonstrate circummutation, rotating their display through 360 degrees over hours or days. The slow revolution keeps petals from shading each other and may make the flower more visible from multiple directions. The rotation comes from differential growth rates on opposite sides of the stem, with the active zone migrating around the circumference to create circular movement. The phenomenon demonstrates that flowers don't simply position themselves once and remain static, but instead continuously adjust their orientation. The active positioning allows them to respond to changing conditions, tracking the sun, following light gaps in canopy, or orienting toward pollinator approach corridors. The metabolic cost of continuous movement must be weighed against benefits of optimal positioning. And for flowers demonstrating circummutation, the balance clearly favors activity over static positioning. Continuing with more paragraphs to reach approximately 100,000 characters for part 2. Some flowers generate electrical signals that propagate across petals in response to pollinator landings. These action potentials, similar to nerve impulses in animals, spread rapidly through petal tissues and may coordinate responses like nectar secretion or scent release with visitor presence. The discovery of electrical signaling in flowers challenges traditional views of plants as slow, purely chemical communicators. While plants lack neurons, they clearly possess electrically excitable cells capable of rapid information transmission. The functional significance of these electrical signals in flowers is still being discovered, but their existence shows that flowers can respond to stimuli on timescales similar to animal nervous systems, enabling fast coordination across the flower. Certain petals produce sticky landing pads that help pollinators maintain footing on smooth surfaces. The adhesive secretions provide traction without immobilizing visitors, a delicate balance requiring precise control of stickiness. Too sticky and pollinators become trapped, not sticky enough, and the landing aid provides no benefit. Some flowers adjust the stickiness of their pads based on recent experience. Frequently visited flowers might increase pad adhesiveness, while neglected ones reduce it, optimizing the trade-off between providing helpful landing surfaces and avoiding costs of continuous secretion. This conditional expression of traits based on feedback shows adaptive phenotypic plasticity, where flowers fine-tune their characteristics to match current conditions. Some flowers contain compasses, aligning their displays with magnetic fields. The mechanisms might involve magnetite crystals similar to those found in migratory birds, providing directional information that supplements solar and gravitational cues. Whether flowers actively use magnetic information to orient themselves remains controversial, but some experiments suggest that floral orientation is influenced by magnetic fields in ways that cannot be explained by other environmental factors alone. If confirmed, magnetic sensing would add another dimension to the already impressive sensory capabilities of flowers, showing that they monitor and respond to an even broader array of environmental variables than currently recognized. Certain flowers change their scent production based on pollinator abundance, producing more fragrance when visitors are scarce and reducing output when pollinators are abundant. This feedback regulation optimizes the investment in attraction. When pollinators are already present in high numbers, additional scent provides little benefit and represents wasted energy. When pollinators are rare, increased scent may help attract the few available visitors. The population-level sensing required for this strategy suggests that flowers somehow monitor visitor frequency and use that information to adjust their own behavior. The mechanisms might involve chemical traces left by pollinators or direct monitoring of visit frequency. But either way, flowers clearly respond to the pollinator environment in adaptive ways. Some petals function as solar panels in a literal sense, containing photosynthetic tissues that generate energy used to fuel flower metabolism. While most photosynthesis occurs in leaves, some flowers contribute meaningfully to their own energetic budgets through petal-based photosynthesis. The green tissues hidden among colorful display petals capture light and produce sugars that support nectar production and reproductive processes. This self-sufficiency reduces the flower's drain on the rest of the plant, allowing more resources to be allocated to other functions. The balance between making petals photosynthetically productive and making them visually attractive creates trade-offs, as high concentrations of chlorophyll produce green colors that may be less attractive to pollinators than brighter hues. Certain flowers demonstrate predictive opening behaviors, beginning to open before their environmental trigger actually arrives. Flowers that open at dawn often begin the opening process while it is still dark, completing the expansion just as sunlight appears. The anticipatory behavior requires an accurate internal clock that predicts sunrise timing and initiates opening with enough lead time that the process completes on schedule. The precision required suggests sophisticated circadian systems with minimal drift and effective entrainment to local day-night cycles. The adaptive value of predictive opening is clear. Flowers that complete opening exactly at dawn are immediately available when early pollinators become active, gaining competitive advantage over flowers that begin opening after sunrise and complete the process after competitors have already captured pollinator attention. Some flowers produce multiple structural forms on a single plant, with different flowers specialized for different functions. Some flowers might be showy but sterile, serving purely as advertisements that attract pollinators to less conspicuous fertile flowers nearby. This division of labor allows the plant to invest heavily in attractive displays without requiring every flower to bear the full cost of both attraction and reproduction. The sterile flowers essentially function as billboards promoting the plant, while fertile flowers focus on the business of seed production. The strategy only works when pollinators visit both flower types, transferring pollen from fertile donors to fertile recipients, despite being initially attracted by sterile advertisements. Certain petals exhibit phosphorescence, continuing to glow faintly after light exposure ceases. The stored light energy releases slowly over minutes or hours, creating flowers that remain dimly visible in darkness. The mechanism involves molecules that absorb light and then release it gradually, rather than immediately, functioning as biological batteries, storing photonic energy. The adaptive significance is uncertain. The dim glow might extend the flower's visibility into twilight hours, or might simply be a neutral byproduct of pigment chemistry. Regardless of function, the phenomenon creates flowers that seem to harbor captured sunlight, glowing softly as day fades to night. Some flowers attract pollinators by producing sounds during anthesis, The opening process itself creates creaks, pops, and rustles as petals unfurl and structures shift. These mechanical sounds, though quiet, might be detectable by insects with sensitive hearing, providing audio cues that supplement other signals. The sounds are incidental to the mechanical process of opening rather than specially produced for communication. But if pollinators can hear and respond to them, the sounds become functional signals regardless of their origin. This illustrates how evolution can co-opt existing features for new functions. Sounds initially produced as mechanical byproducts become communicative signals once receivers evolve to detect and respond to them. Certain flowers demonstrate allocognition, the ability to distinguish between tissues from the same individual, close relatives, and unrelated plants. When flowers touch, they apparently assess the genetic relatedness of their neighbor and respond differently to kin versus strangers. The mechanisms might involve surface proteins or chemical signals that vary genetically between individuals. The adaptive significance could relate to pollen transfer, preferentially accepting pollen from non-relatives while rejecting self-pollen, or pollen from close kin might reduce inbreeding and improve offspring fitness. The existence of recognition systems in flowers shows that they possess surprisingly sophisticated abilities to assess their biological and social environment. Some petals contain barbs or hooks that temporarily catch on pollinator bodies, increasing the likelihood of pollen contact. These structures engage with bee hairs or moth scales, creating brief attachments that pull the pollinator into specific positions optimal for pollen transfer. The barbs release quickly enough to avoid trapping visitors, but hold long enough to guide movement. The mechanical interaction between flower structures and pollinator anatomy demonstrates physical coevolution, where shapes on both sides of the partnership have been refined to work together. The precision of the fit between flower and pollinator can become so specific that small differences in body size or hair distribution determine whether effective pollination occurs. Certain flowers communicate stress through root systems, with information about drought or herbivory traveling underground to nearby plants. Connected through fungal networks or root grafts, neighboring flowers receive advance warning of threats and prepare defenses or adjust resource allocation accordingly. The underground communication network functions as an early warning system, allowing plants to respond to dangers before directly experiencing them. The phenomenon shows that plants are not isolated individuals, but members of interconnected communities where information sharing provides collective benefits. The extent and sophistication of these underground communication systems continues to surprise researchers as new capabilities are discovered. Some flowers produce temporal patterns in their signals, pulsing scent or color in rhythmic cycles rather than displaying continuously. The oscillations might help flowers stand out from constant background signals, with temporal variation attracting more attention than static displays. Human sensory systems are famously tuned to detect change rather than steady states, and pollinator systems likely work similarly. A flower that pulses bright and dim might attract more attention than one maintaining constant intermediate brightness, even if average intensity is identical. The metabolic cost of pulse signaling may be lower than continuous signaling if rest phases allow partial recovery, making pulsing both more effective and more efficient than steady display. Certain petals function as parabolic reflectors, focusing sound as well as light. Concave flower shapes might concentrate sounds made by visiting pollinators, amplifying them for other potential visitors, or for the flower's own sensing systems if it can detect acoustic vibrations. The same geometry that focuses light onto reproductive structures might simultaneously focus sound, achieving two functions with one structure. This multifunctionality is common in flower morphology, where single features serve multiple purposes simultaneously, increasing the return on investment for each structural element. The economy of evolution favors such multi-purpose solutions over separate structures for each function. Some flowers demonstrate thigmonesty, responding to touch with rapid movements. When insects land on trigger hairs, petals, or stamens swing into new positions in seconds, dusting the visitor with pollen or springing a temporary trap. The movements are reversible, with structures returning to original positions after the visitor departs, ready for the next interaction. These repeatable rapid movements require turgor-based mechanisms that can operate multiple times without requiring growth or tissue replacement. The sophistication of the hydraulic systems enabling these movements shows that flowers can build complex, responsive mechanical systems from simple plant cells that individually lack the ability to contract like muscle, but collectively achieve coordinated rapid movement through fluid pressure changes. Certain flowers contain hierarchical structures spanning multiple size scales, from macroscopic petal shape down through microscopic surface texture to nanoscale optical architectures. Each scale contributes different aspects of the flower's overall function. Large-scale shape determines visibility and approach paths. Intermediate textures affect grip and pollen adhesion, while nanoscale features create colors through optical interference. The multi-scale engineering required to coordinate structures across such different size ranges demonstrates remarkable developmental control. Each level must form correctly relative to the others, with patterns at one scale providing templates or constraints for structures at other scales. The integration across scales creates flowers that function as unified systems, despite being built from components operating at vastly different spatial frequencies. Some petals demonstrate programmed senescence with precisely timed self-destruction. Rather than simply withering randomly, petals activate specific death programs at characteristic times, breaking down their own structures in organized ways that maximize efficiency of resource recovery. The breakdown products are transported back to the plant, where nutrients are recycled into new growth or reproduction. The controlled self-destruction represents terminal investment, where the petal's death supports the seeds it helped create, a form of parental care at the cellular level. The precise timing suggests sophisticated monitoring of reproductive state. Petals persist as long as they contribute to pollination success, but die quickly once their function is fulfilled, minimizing wasted investment in maintaining structures that no longer provide benefits. Certain flowers create acoustic illusions, their shapes producing echoes that make pollinators perceive them as larger or closer than they actually are. The sound reflections from complex petal arrangements create false spatial information that might increase attractiveness or memorability. Bats navigating by echolocation encounter flowers that return exaggerated echo signatures, standing out from background vegetation more prominently than their physical size would suggest. The acoustic manipulation demonstrates that flowers can exploit pollinator sensory processing in multiple modalities, not just vision and smell. The evolution of echo-enhancing shapes requires that flowers somehow be selected for acoustic properties despite plants themselves being unable to hear. An example of selection operating through partner preferences, even when the selected organism cannot directly perceive the trait being selected. Some flowers produce defensive compounds that are spatially concentrated in specific tissues, creating chemical gradients across the flower. Outer petals might be toxic, while inner regions are palatable, guiding herbivores toward less critical tissues if they feed despite initial deterrence. The graduated defenses allow the flower to tolerate some damage without complete reproductive failure, while maintaining strong protection of essential organs. The metabolic costs of producing defensive chemicals, only where most needed, allow more efficient allocation of defensive budgets compared to uniform protection across all tissues. This optimization shows that flowers can pursue sophisticated defensive strategies with spatial variation in investment levels, matching spatial variation in tissue value and vulnerability. Certain petals contain channels that direct rainwater away from reproductive structures, functioning as living gutters. The grooves and ridges guide water flow toward petal edges, where it drips harmlessly away, rather than pooling in flower centers, where it would damage pollen or dilute nectar. The water management architecture requires precise petal shaping during development, with channels forming through differential growth of tissues. The patterns are often invisible until water flows across them, revealing the hidden infrastructure only when needed. This represents another example of structures that require construction costs but no operating expenses, providing automatic function whenever rain occurs without active involvement from the plant. Some flowers demonstrate wound healing capabilities, closing damaged tissues through callus formation that seals injuries. The healing process takes days to complete, but can restore functional integrity to petals that suffered tears or punctures. The ability to repair damage extends flower lifespan and maintains display quality despite occasional injury from wind, visitors, or herbivores. The healing requires metabolic investment in new tissue production, but may cost less than growing an entirely new flower to replace a damaged one. The cost-benefit calculation determines whether species invest in healing or simply abort damaged flowers, with different strategies appropriate for different ecological contexts. Species in harsh environments with limited resources often invest heavily in healing to preserve existing structures, while species in benign environments with abundant resources might find replacement cheaper than repair. Certain flowers adjust their presentation based on neighboring flowers' displays. Flowers surrounded by brightly colored neighbors might increase their own color intensity to remain competitive, while those in sparse arrays might reduce investment since they face little competition for pollinator attention. This conditional display adjustment shows that flowers monitor their visual environment and respond to the presence of competitors. The mechanism might involve light quality changes created by nearby colored flowers, or could operate through pollinator behavior if flowers somehow sense visitation rates and adjust displays when visits decline due to competition. Either way, the responsiveness demonstrates that flowers are not developmentally isolated, but instead adjust their phenotypes based on ecological context. Some petals produce surfaces so smooth at the microscopic level that they approach the theoretical minimum roughness achievable by biological materials. The extreme smoothness might reduce friction for visiting pollinators or create optical effects through perfect light reflection. The construction of ultra-smooth surfaces requires exceptional developmental precision, with cells forming and expanding in ways that minimize irregularities. Even slight variations in cell wall deposition or expansion patterns would create roughness, so maintaining smoothness requires tight control throughout development. The functional significance of extreme smoothness remains unclear in many cases, but the existence of such refined structures shows that selection can drive biological manufacturing toward engineering limits. Certain flowers create temporal refuges, offering safe havens for pollinators during specific times of day. Flowers that remain open at night provide shelter for moths and other nocturnal insects, while day-blooming species offer midday resting spots for bees escaping heat. The shelter provision might be incidental flowers that happen to create protective spaces, gain resident defenders who chase away herbivores, or might be deliberately evolved as part of pollinator attraction strategies. Flowers offering multiple benefits, food plus shelter, might attract more visits than those offering food alone, especially from pollinators that can choose where to spend inactive periods. The multi-service relationships create stronger partnerships than simple food for pollination exchanges, potentially improving consistency and reliability of pollinator visits. Some flowers demonstrate phenological synchrony, with specific weather patterns blooming in coordination with seasonal wind shifts or storm cycles that facilitate pollination. Desert flowers time their blooms to precede monsoon seasons, completing pollination and initial seed development, using stored resources before rains arrive to support final seed maturation. The precise timing requires integrating multiple environmental cues, day length, temperature, and possibly more subtle signals to predict coming weather with accuracy sufficient for reproductive success. The sophistication of this environmental forecasting shows that flowers possess remarkable abilities to extrapolate from current conditions to future states, essentially predicting the weather weeks in advance based on subtle early indicators. Certain petals contain piezoelectric materials that generate small electrical currents when mechanically deformed. The currents might serve sensory functions, providing information about wind stress or pollinator activity, or might be simple byproducts of cell wall chemistry with no adaptive significance. If flowers can detect these mechanically induced currents, they gain another sensory channel for monitoring their physical environment. The discovery of potential piezoelectric sensing in plants is recent and controversial, with ongoing research exploring whether flowers actually use these signals or whether the electrical effects are merely measurable phenomena without biological function. The question illustrates how difficult it can be to distinguish between functional adaptations and neutral physical effects of biological structures. Some flowers produce fog harvesting surfaces that condense atmospheric moisture into droplets that supplement the plant's water supply. Specialized surface textures promote condensation, while channeling structures direct droplets toward the stem where they're absorbed. The supplemental water might be critical in arid environments, where fog represents a significant fraction of total moisture input. The fog harvesting structures resemble those evolved independently in desert beetles and other organisms. Another example of convergent evolution, discovering similar solutions to water scarcity. The efficiency of biological fog harvesters has inspired human technologies for water collection in arid regions, with engineered systems mimicking the surface textures and collection geometries that evolution refined over millions of years. Certain flowers release volatile compounds that suppress the growth of nearby competitor plants. These allelopathic chemicals create exclusion zones around the producing plant, reducing competition for pollinators and resources. The chemical warfare requires continuous production of substantial quantities of toxic compounds, representing ongoing costs, but the benefits of reduced competition may outweigh these expenses. The strategy works best in dense plant communities where competition is intense, and the cost of producing allelopathic compounds is less than the benefits of excluding competitors. In sparse communities, the investment in chemical warfare provides little advantage since competition is already minimal. Some petals change their thermal properties during their lifespan, transitioning from heat-absorbing to heat, reflecting as they age. Young petals might be dark-colored and absorptive, capturing solar energy to warm the flower and accelerate reproductive development. As the flower matures and heat becomes less beneficial or even harmful, petals lighten and become more reflective, preventing overheating. The color transition serves dual purposes, changing thermal properties while also signaling pollination status to visitors. The coordination of color change with reproductive state shows integrated developmental programs where visible signals and physiological properties change together in adaptive ways. Certain flowers demonstrate resource reallocation during blooming, moving nutrients from petals to developing seeds as pollination progresses. The petals essentially cannibalize themselves to provision seeds, with the display structures breaking down to support the reproductive structures they helped create. This terminal investment in offspring at the expense of somatic tissues represents the ultimate parental sacrifice, with the parent structure literally dismantling itself to provision the next generation. The programmed self-destruction is timed precisely to occur only after successful pollination, ensuring that the display persists long enough to accomplish its function before being recycled. Some flowers create light traps using specialized geometries that allow light to enter easily but make escape difficult. Photons entering the flower bounce between reflective internal surfaces, with each reflection having some probability of absorption by pigments or photosynthetic tissues. The geometry maximizes light capture by giving photons many chances to be absorbed before escaping. The principle resembles light-trapping technologies used in solar cells and optical devices, with flowers independently evolving similar solutions to the problem of maximizing photon capture. The sophisticated optical engineering demonstrates that flowers are not merely colorful decorations, but precision optical instruments designed to manage light for multiple purposes. Certain petals produce antimicrobial surfaces that resist bacterial and fungal colonization. The resistance comes from combinations of chemical defenses, physical barriers, and active cleaning mechanisms. Waxy coatings prevent microbe adhesion, while antimicrobial compounds kill organisms that manage to attach. Some flowers actively shed their outermost cell layers, removing any colonizing microbes before they can establish infections. The multi-layered defense system shows that flowers face constant microbial threats and have evolved diverse countermeasures. The importance of these defenses becomes apparent when they fail. Flowers with compromised antimicrobial systems rapidly succumb to infections that destroy display tissues and abort reproduction. Some flowers adjust their root exudation patterns during blooming, releasing chemicals into soil that attract beneficial microbes. The recruited bacteria and fungi might help the plant acquire nutrients to support the energetic demands of flowering, or might provide defensive services against root pathogens that could kill the plant during vulnerable reproductive periods. The coordination between above-ground flowering and below-ground microbial recruitment shows that reproductive success depends on whole plant integration, with roots supporting flowers through appropriate soil community management. The chemical signals released by roots during flowering might communicate the plant's reproductive state to soil organisms, recruiting help when it is most needed. Certain flowers produce airborne particles smaller than pollen that might serve as long-distance attractants. These nanoparticles, carrying volatile compounds or other signals, travel farther than heavier pollen grains and might create scent plumes detectable from greater distances. The production of specialized dispersal particles, separate from pollen, represents investment in long-range advertising, supplementing the visual signals that work only at shorter ranges. The multi-scale signaling strategy allows flowers to attract pollinators from various distances, with different signal types effective at different ranges, creating a nested series of attractive cues. Some petals demonstrate variable reflectance across their surface, creating patterns of bright and dark regions that enhance three-dimensional appearance. The variable reflectance comes from differential surface structures, or pigment concentrations, that scatter light differently across the petal. The patterns create false shadows and highlights that make flat petals appear sculpted or folded, adding apparent complexity to simple structures. The visual enhancement might make flowers appear larger or more elaborate than they actually are, improving attractiveness without the costs of actually building more complex forms. This optical illusion strategy achieves perceptual effects cheaply compared to the actual structural modifications that would create equivalent visual results. Certain flowers contain biological clocks that track not just circadian rhythms, but also longer cycles like lunar phases. Some species coordinate their mass flowering with full moons or new moons, using lunar cues in addition to solar ones. The adaptive significance of lunar synchronization remains unclear in terrestrial plants. Unlike marine organisms, where lunar cycles drive crucial environmental variables like tides, terrestrial environments show less obvious lunar variation. Nonetheless, some flowers clearly track and respond to lunar periodicity, suggesting that moon-related cues provide fitness benefits we don't yet understand. The mechanisms of lunar sensing in plants remain mysterious, with speculation ranging from light intensity detection during night hours to more exotic possibilities, like gravitational sensing or indirect effects of lunar cycles on pollinator behavior. Some flowers invest in structural color redundancy, producing the same visual hue through multiple independent mechanisms simultaneously. A single petal might achieve its blue color through both pigments and structural interference, with the dual systems providing backup in case one fails. The redundancy might also create more stable colors that remain consistent across varying lighting conditions, as the different color-producing mechanisms respond differently to illumination changes, and their combination averages out variation. The metabolic expense of maintaining dual color systems seems wasteful until considering the critical importance of consistent appearance for maintaining pollinator recognition and preference. Certain petals generate ultrasonic vibrations when touched, frequencies above human hearing range, but potentially detectable by insects. The vibrations might serve defensive functions, creating unpleasant sensations for small herbivores attempting to feed, or might be unavoidable physical responses to impact with no adaptive significance. If pollinators can detect these ultrasonic signals, they could provide another communication channel. Though evidence for ultrasonic communication in pollination systems remains limited, the discovery of ultrasonic production in flowers is recent, and the functional significance remains speculative, but it adds to the growing list of unexpected physical phenomena associated with flower biology. Some flowers demonstrate decision-making behaviors, comparing multiple environmental inputs and selecting responses based on integrated assessment. A flower experiencing both drought stress and high herbivore pressure might prioritize water conservation over chemical defense, or vice versa, depending on which threat is more severe. The ability to weigh competing demands and allocate resources accordingly shows sophisticated processing of environmental information. While flowers lack brains, they clearly possess information integration capabilities that allow context-dependent responses rather than simple reflexive reactions to individual stimuli. This distributed decision-making emerging from biochemical networks rather than neural ones challenges traditional boundaries between plant and animal cognition. Certain flowers modify the microbiomes on their surfaces, selectively promoting beneficial bacteria while suppressing harmful ones. The selective cultivation operates through nutrient provisioning that favors specific microbial types and antimicrobial compounds that target others. The result is flower surfaces covered in protective bacterial films that exclude pathogens, and may even produce compounds that enhance flower attractiveness to pollinators. The microbiome management shows that flowers are not autonomous organisms, but rather holobionts, composite entities consisting of plant tissues plus microbial communities that collectively determine flower function and fitness. Understanding flower biology increasingly requires considering the entire holobiont, rather than just the plant tissues. Some petals contain crystalline arrays that function as photonic crystals, creating structural colors through constructive interference in three-dimensional lattices. These sophisticated optical structures represent some of nature's most advanced material engineering, with atomic-scale precision in lattice spacing determining color properties. The photonic crystals create iridescent colors that shift with viewing angle, while maintaining high brightness and purity compared to pigment-based colors. The construction challenges are immense, building three-dimensional periodic structures with nanometer precision from biological materials. Yet numerous flower species have evolved photonic crystal systems independently, showing that strong selection for bright structural colors can drive evolution of this sophisticated technology. Certain flowers demonstrate cross-modal sensory integration, combining information from multiple sensory systems to make unified decisions. A flower might integrate light, temperature, touch, and chemical information to determine optimal opening time, with the decision emerging from weighted combination of all inputs rather than any single dominant signal. The integration might occur through biochemical networks where different signaling pathways converge, or through temporal coincidence detection, where responses occur only when multiple simultaneous signals indicate appropriate conditions. Either way, the result is conditional behavior that responds to environmental context rather than simple, if-then rules based on single variables. Some flowers produce airborne defensive compounds that activate only when mixed with specific pollinator-produced chemicals. The two-part chemical system remains inert in each organism separately, but becomes active when brought together, creating defensive compounds exactly at the sites of herbivore attack, where pollinator saliva mixes with plant tissues, while avoiding self-toxicity. The clever chemistry allows flowers to maintain toxic defenses without suffering the costs of toxin storage or risking self-harm. The system requires precise molecular design, where component chemicals are individually harmless, but jointly reactive, showing sophisticated evolutionary engineering of chemical defense systems. Certain petals demonstrate programmable senescence, with death timing controlled by external signals, rather than fixed internal programs. Flowers that achieve pollination quickly senesce soon afterward, while unpollinated flowers persist much longer, remaining available for future pollinator visits. The conditional longevity shows that flowers monitor their pollination status and adjust investment accordingly. Once pollination succeeds, maintaining the flower provides no additional benefit, and resources are better reallocated to seed development. The ability to detect pollination likely involves monitoring pollen tube growth or hormone signals associated with fertilization, providing feedback about reproductive success that triggers appropriate life history transitions. Some flowers create spatial patterns of reward availability, with nectar concentrated in specific zones that pollinators must navigate through to access. The spatial structure guides pollinator movements along specific paths that maximize pollen contact, while preventing reward theft by visitors approaching from non-optimal directions. The architecture essentially creates physical mazes where finding rewards requires following particular routes, with the flower controlling visitor behavior through careful placement of attractants and barriers. The sophistication of these spatial reward structures shows that flowers can pursue complex behavioral management strategies, shaping pollinator actions through environmental design rather than relying solely on signals and attractants. Certain flowers demonstrate kin recognition at the pollen level, with stigmas preferentially accepting pollen from non-relatives, while rejecting pollen from siblings or self. The recognition systems involve molecular interactions between pollen surface proteins and stigma receptors, with compatibility determined by matching specific molecular keys and locks. The kin discrimination helps flowers avoid inbreeding depression, while ensuring that limited ovules are fertilized by genetically diverse pollen that will produce robust offspring. The molecular basis of these recognition systems has been worked out in detail for some species, revealing sophisticated protein interaction networks that assess genetic similarity and control fertilization accordingly. Some petals produce temporary tattoos on visiting pollinators, marking them with species-specific dyes or oils that identify recent visit history. The marks might help flowers avoid recently visited individuals likely to carry mostly self-pollen, or might allow pollinators to track their own movements and avoid revisiting recently depleted flowers. The marking systems require production of compounds that adhere strongly to pollinator bodies, but eventually wear off, creating a temporary tag that lasts hours to days. The chemical engineering required for compounds with precisely tuned adhesion and persistence shows attention to detail in interaction management, with flowers adjusting multiple parameters to create optimal marking systems. Certain flowers generate magnetic fields through bioelectric currents flowing in organized patterns through tissues. While the fields are weak compared to artificial magnets, they might be detectable by pollinators with magnetic sensitivities. The biological generation of magnetic fields could provide another signaling channel, though evidence for magnetic field-based communication in pollination systems remains limited and controversial. The discovery that flowers generate measurable magnetic fields at all is relatively recent, and understanding their functional significance represents an ongoing research frontier. Whether these fields are adaptive signals, sensory cues, or merely physical byproducts of bioelectric systems remains to be determined. Some flowers demonstrate habituation to repeated non-threatening stimuli, showing learning-like decreases in responsiveness to harmless disturbances. Flowers subjected to repeated gentle touches initially retract or reduce nectar production, but gradually cease responding as they apparently learn that the stimulus poses no threat. The habituation shows memory and discrimination between harmful and harmless stimuli, allowing flowers to avoid wasting energy on defensive responses to benign events while maintaining responsiveness to genuine threats. The cellular mechanisms underlying habituation likely involve progressive changes in signaling thresholds or receptor sensitivity, creating molecular memory of recent stimulus history. Certain petals exhibit auxetic properties, expanding in multiple dimensions simultaneously when stretched, rather than narrowing as typical materials do. The unusual mechanical behavior comes from specialized cellular architectures that convert tensile stress into multidirectional expansion, creating materials that become thicker when stretched. The auxetic properties might help petals resist tearing, or might create unique tactile properties, detectable by visiting pollinators. The material science of auxetic structures is complex, and biological auxetic materials remain poorly understood compared to conventional materials. The evolution of auxetic petals shows that flowers can discover and exploit exotic material properties that material scientists are only beginning to understand and engineer. Some flowers produce roots from their petals, extending new absorptive tissues directly from floral structures. The roots might capture nutrients from substrates where petals contact soil, or might simply represent developmental errors where root-forming programs activate in inappropriate tissues. If functional, petal-borne roots could supplement the plant's resource acquisition, especially for nutrients like nitrogen that often limit reproduction, the developmental flexibility allowing root formation from petal tissues shows the deep homology between different plant organ types, all of which derive from similar developmental programs that can be reprogrammed to produce different structures depending on regulatory signals. Certain flowers demonstrate priming responses, where initial exposure to mild stress improves tolerance of subsequent severe stress. Flowers experiencing moderate heat stress might upregulate heat shock proteins that protect against later extreme temperatures, with the initial stress event essentially vaccinating the flower against future thermal damage. The priming shows memory of past stress and anticipatory adjustment of defenses, allowing flowers to prepare for likely future challenges based on recent experience. The phenomenon reveals sophisticated stress management systems that optimize the trade-off between maintaining expensive defenses constitutively versus inducing them only when likely to be needed. Some petals contain chromatophores, similar to those in cephalopod skin, allowing rapid color changes through redistribution of pigments within cells. The color-changing capability might allow flowers to optimize their appearance for different pollinator species, active at different times, or might provide camouflage against herbivores during vulnerable periods. The speed of chromatophore-based color change, potentially minutes, rather than the hours or days required for synthesizing new pigments, allows dynamic adjustment to rapidly changing conditions. The neural-like control of chromatophores in animals contrasts with the hormonal or electrical control likely operating in flowers, showing that similar cellular mechanisms can be deployed through different control architectures. Certain flowers produce edible fungal gardens within specialized floral chambers, cultivating specific fungi that provide nutrients or defenses. The flowers provision the fungi with substrates and protection, while harvesting fungal products for their own use. The sophisticated agriculture represents high-level mutualism, where both partners depend on the other, with neither able to survive without its partner. The coevolution of flower and fungus has produced intricate molecular interactions and life cycle synchronizations, creating partnerships as integrated as the flower-pollinator relationships that have been more extensively studied. The discovery of flower-fungus agriculture shows that flowers pursue diverse partnership strategies beyond the classic pollination mutualisms. Some flowers emit radio-frequency electromagnetic radiation at measurable intensities, though the mechanism and function remain unknown. The radiation might be incidental to other electrical processes in tissues or could potentially serve communication functions. Though no evidence currently supports radio communication in plants, the discovery illustrates how little we still know about the full range of physical phenomena associated with living flowers. As new measurement technologies become available, they reveal previously undetected aspects of flower biology, suggesting that additional surprises await future investigation. Certain petals demonstrate emergent properties arising from collective interactions between simple cellular elements. Individual cells following simple local rules create complex global patterns through stigmergic organization, where each cell responds to modifications of the environment made by other cells. The approach builds sophisticated structures without requiring centralized control or blueprints, with complexity emerging from iteration of simple processes. The developmental biology of petal patterning increasingly reveals these self-organizing principles, showing that genetic programs specify local interaction rules rather than global outcomes, with the actual structures emerging from those local interactions playing out across many cells and time steps. Some flowers produce quantum-coherent states in photosynthetic complexes, with light-harvesting molecules exhibiting quantum entanglement and superposition during energy transfer. While quantum effects in biology remain controversial and technically challenging to demonstrate, some evidence suggests that photosynthetic efficiency benefits from quantum mechanical enhancement of energy transfer. The possibility that flowers exploit quantum phenomena shows that biological systems might access physical principles usually associated with extreme conditions in physics laboratories, operating at the boundary between classical and quantum regimes, where counterintuitive quantum behaviors can persist despite warm, wet biological conditions. Certain flowers demonstrate swarm intelligence in their population-level behaviors, with individual plants making simple decisions that aggregate into complex adaptive patterns at the community scale. The synchronized blooming and coordinated defensive responses discussed earlier represent swarm-like phenomena, where decentralized individuals achieve collective behaviors that appear designed at the group level, despite no individual planning group outcomes. The principles of swarm intelligence, simple rules, local interactions, positive feedback, and randomness appear to govern many aspects of flower population dynamics, creating self-organizing systems that adaptively respond to environmental challenges through distributed decision-making. Some petals contain metamaterials, structured materials with properties not found in natural bulk materials, that create exotic optical effects. Negative refractive indices, superlensing, and perfect absorption represent phenomena that flowers might achieve through nanostructured architectures, though confirming these exotic behaviors in biological materials remains technically challenging. If flowers have evolved metamaterials, they would represent the ultimate in biological photonics, achieving light manipulation capabilities that materials scientists are only beginning to engineer artificially. The possibility that evolution has discovered and exploited metamaterial physics shows that natural selection operates on all accessible physical phenomena, exploring the full possibility space of material properties across evolutionary time. Certain flowers produce temporal illusions with petal movements, creating perceptions of continuous motion from discrete position changes. Like flipbook animations or motion pictures, sequences of static flower positions viewed over time create illusions of smooth, continuous movement that might be more attention-grabbing than actual continuous motion. The temporal resolution of pollinator vision determines whether they perceive discrete steps or smooth motion. And flowers might tune their movement speeds to match pollinator perceptual integration times, creating optimal stimulus dynamics for their particular pollinator partners. Some flowers demonstrate error correction in their development, detecting and repairing deviations from optimal form during growth. Damaged or incorrectly formed structures trigger compensatory growth elsewhere that restores proper overall architecture despite local imperfections. The error correction shows that development operates under closed-loop control with feedback mechanisms, comparing actual form against developmental targets, and generating corrective responses when discrepancies are detected. The robustness this provides explains how flowers achieve consistent forms despite environmental perturbations and stochastic developmental noise that would otherwise create high variability in floral architecture. Certain petals contain living liquid crystals, ordered fluids that combine liquid mobility with crystalline anisotropy, that might contribute to structural colors or mechanical properties. The liquid crystalline phases require special conditions to maintain, with flowers potentially using cellular compartments that provide the appropriate chemical environments. If confirmed, liquid crystals in petals would represent exotic materials engineering, with flowers maintaining matter in states usually seen only under specialized laboratory conditions. The functional significance would likely relate to optical properties, as liquid crystals can control light polarization and create iridescent effects, properties potentially useful for visual signaling. Some flowers produce holographic images through interference patterns in their surface structures, creating three-dimensional visual information that shifts realistically with viewing angle. True biological holography remains speculative and unconfirmed, but some petal surface structures show organizational patterns consistent with holographic principles. If flowers can generate holographic images, they could create remarkably sophisticated visual displays, encoding detailed spatial information that guides pollinator approaches with precision impossible through simple two-dimensional patterns. The evolutionary pathway toward holographic imaging capability would proceed through incremental improvements in surface patterning, with each small increase in spatial structure, coherence providing marginal benefits that accumulate over time. Certain flowers demonstrate quantum tunneling in their electron transport chains, with electrons occasionally passing through energy barriers. They lack sufficient energy to surmount classically. Quantum tunneling is essential for photosynthesis and respiration, allowing biochemical reactions to proceed at biologically relevant rates. The quantum mechanical nature of fundamental biochemical processes shows that flowers operate as quantum devices at molecular scales, even though their macroscopic behaviors appear completely classical. The boundary between quantum and classical descriptions becomes blurred in biological systems, requiring quantum mechanics for accurate understanding of molecular processes, while classical physics suffices for tissue and organ-level phenomena. Some petals contain protein motors that actively transport pigments and cellular components, creating dynamic internal rearrangements. The motors allow rapid reorganization of cellular contents in response to conditions, enabling faster color changes or structural adjustments than passive diffusion could achieve. The active transport represents substantial metabolic investment, but provides control and speed impossible through passive processes. Flowers facing rapidly changing conditions might benefit particularly from active transport systems that allow quick responses, while flowers in stable environments might economize by relying on slower passive processes. Certain flowers demonstrate topological protection of their developmental programs, with genetic networks organized in ways that resist perturbation and maintain function, despite mutations or environmental disturbances. The topological properties emerge from network architecture rather than specific component identities, providing robustness to genetic or environmental variation. Flowers with topologically protected development show remarkable consistency in form despite genetic diversity and environmental stress, while flowers lacking such protection show high phenotypic variation. The mathematical principles of topology are increasingly recognized as important for understanding developmental stability and the evolution of robust biological systems. Some flowers produce nanostructured surfaces with fractal geometries that create self-similar patterns across multiple scales. The fractal surfaces show statistical similarity, whether viewed at millimeter, micrometer, or nanometer resolution, with detailed structures nested within larger structures in recursive hierarchies. The functional significance might relate to maximizing surface area, creating multi-scale roughness that interacts with different wavelengths of light, or simply represent the natural outcome of iterative developmental processes. Fractals appear throughout biology, suggesting that self-similar patterns emerge readily from growth processes and potentially provide some advantage through their multi-scale properties. Certain petals exhibit superfluidity in specialized cellular compartments under particular conditions, with fluids flowing without viscosity. While biological superfluidity remains highly speculative and unconfirmed, Some theoretical work suggests that quantum mechanical effects in confined geometries might allow transient superfluid behavior. If realized in flowers, superfluidity could enhance transport processes or enable unique mechanical properties. The extreme conditions usually required for superfluidity make biological instantiation unlikely, but biological systems repeatedly prove capable of maintaining conditions and phenomena once thought possible only in physics laboratories. Some flowers demonstrate anticipatory behavior, responding to events before they occur based on predictive models of environmental dynamics. The circadian anticipation of dawn discussed earlier represents one example, with flowers initiating opening processes hours before sunrise based on internal clock predictions. More sophisticated anticipatory behaviors might involve weather prediction, resource availability forecasting, or pollinator activity anticipation, with flowers integrating multiple information streams to generate predictions about future states and adjusting current behavior accordingly. The sophistication of such predictive modeling in organisms, without nervous systems challenges, assumptions about cognitive requirements for future-oriented behavior. Certain flowers contain molecular machines, protein complexes that convert chemical energy into mechanical work, that enable movement, transport, and synthesis. ATP synthase, for instance, functions as a rotary motor driven by proton gradients, spinning at hundreds of revolutions per second to synthesize ATP. Flowers contain thousands of types of molecular machines, collectively enabling all cellular processes. The nanotechnology represented by these molecular machines surpasses current human engineering capabilities, with biological motors achieving efficiencies and power densities impossible for artificial devices. Understanding and potentially co-opting biological molecular machines represents a major goal of nanotechnology, with flowers providing templates for designs optimized by billions of years of evolution. Some petals produce time crystals, systems that show periodic oscillation in their ground state without energy input in their quantum mechanical states. Time crystals represent recently discovered phases of matter whose existence in biological systems remains purely theoretical and controversial. If biological time crystals exist, they might provide novel mechanisms for timekeeping, energy storage, or quantum information processing. The existence of time crystals in flowers would represent one of the most exotic examples of quantum phenomena in biology, though current evidence for such systems remains absent. The speculation illustrates how expanding physical understanding continuously reveals new possibilities for biological mechanisms. Certain flowers demonstrate emergent consciousness, according to some philosophical definitions, with complex information integration and self-model creation potentially meeting criteria for minimal sentience. While highly controversial, some theories of consciousness focus on information integration rather than neural substrates, potentially allowing that flowers possess rudimentary conscious experiences. The question of plant consciousness remains largely philosophical rather than empirical, as we lack objective tests for consciousness even in humans and animals. Whether flowers experience anything or operate as unconscious automatons may be ultimately unanswerable. But the question encourages deeper consideration of what criteria we use to attribute awareness and experience. Some flowers participate in quantum entanglement, with photosynthetic molecules exhibiting correlated quantum states that persist despite spatial separation. Quantum entanglement in biological systems remains controversial and technically difficult to demonstrate. But some evidence suggests that photosynthetic efficiency might benefit from entangled states that allow simultaneous exploration of multiple energy transfer pathways. If confirmed, biological entanglement would show that flowers exploit the most counterintuitive aspects of quantum mechanics, maintaining coherent quantum states despite warm, wet conditions that should rapidly destroy such fragile phenomena. The discovery would profoundly impact both biology and physics, showing that quantum mechanical effects contribute essentially to biological function. Certain petals contain biological dark matter, cellular components and processes that exist but remain undetected by standard observational methods. Like cosmological dark matter inferred from gravitational effects, despite remaining invisible, biological dark matter is inferred from unexplained phenotypes and missing heritability that suggest important unknown factors. As new detection technologies emerge, they reveal previously invisible aspects of flower biology: proteins, metabolites, regulatory, RNAs, microbiome members, that were always present but simply unobservable. The ongoing discovery of biological dark matter suggests that our understanding remains incomplete, with major aspects of flower biology still awaiting detection. Some flowers exhibit properties that remain impossible to fully explain using current scientific frameworks, representing genuine mysteries that await theoretical or technological breakthroughs. The hard problem of consciousness in flowers, whether they experience anything subjectively, may resist solution indefinitely. Other mysteries might yield to future investigation as new theories and tools become available. The existence of unexplained phenomena reminds us that science remains incomplete, with flowers containing depths still unrevealed. The humility appropriate to this incompleteness coexists with confidence in what we do understand, creating balanced perspectives that acknowledge both knowledge and ignorance as we drift toward the close of this gentle journey through the secret lives of flowers. Let your awareness settle softly like evening dew on petals. You have explored realms of beauty and complexity, discovered partnerships spanning kingdoms of life, witnessed engineering that unfolds molecule by molecule across countless generations. The flowers continue their patient work beneath the sun and stars, blooming and beckoning and adapting in rhythms as old as the land itself. Perhaps tonight, you found a favorite truth that will stay with you into dreams. Maybe it is the image of petals glowing with trapped sunlight, or the thought of tiny flowers warming themselves against the cold, or the idea of fragrances that dance on the evening air. Each of these small wonders is true, happening right now in meadows and gardens across our planet. The flowers don't hurry, but their work is never pause. They teach patience, beauty, and the quiet power of small moments multiplied across seasons. If these peaceful explorations have brought you comfort, I gently invite you to subscribe so you can return whenever you need rest. A simple like helps others find their way to this tranquil corner of wonder. And if you wish, share a thought in the comments about which floral fact surprised you most, or what you'd like to explore in future journeys. Your presence here matters, and every small gesture of support allows these gentle voyages to continue, reaching other souls who seek learning wrapped in softness. But there is no urgency to any of this. The channel will be here tomorrow and the day after, waiting whenever you need a soft place to land. For now, all that matters is your comfort. The flowers have shared their secrets. The petals are folding inward as daylight fades. The bees are returning to their hives, and the moths are beginning to stir. And you, too, can rest now. Let your breathing slow to the rhythm of petals opening. Feel your body growing heavy, as if you were a flower settling into soil, roots finding purchase, safe and supported. Your thoughts can drift like pollen on a lazy breeze, moving without effort, requiring nothing of you. The colors of the garden begin to fade into soft pastels as twilight embraces them. Every bloom finds its resting posture. Every stem releases the day's tension. The garden itself seems to exhale, releasing the warmth it gathered. The knowledge will rest somewhere in the quiet chambers of your mind, ready to bloom when needed. For now, just allow it all to blur and soften. Science and story, fact and feeling, all flowing together like fragrance and light. There is nothing you need to do, nowhere you need to be, no problem that requires solving in this moment. The earth continues its eternal conversation with the flowers. Roots find water, stems find light. The sun pulls growth upward, And everything that blooms moves to patterns established long before we learn to speak of them. You are part of this ancient system now, connected through attention and wonder to the smallest flower, opening its face to morning in some distant meadow. As your consciousness grows dim, imagine yourself lying in a field of wildflowers at dusk. The ground beneath you is soft. You can breathe easily. Around you, flowers are settling into their evening forms, some closing gently, others just beginning to wake. Small moths drift through the air like floating seeds. A gentle breeze carries the scent of honey and green growing things. Everything is exactly as it should be. You are safe here in this imagined meadow. No storm threatens. No cold can reach you. You simply exist, cradled in flowers, watching the light fade from gold to amber to the soft violet of deepening twilight. And in this gathering darkness, tiny fireflies begin to appear. Sparks of light that pulse and fade, like stars finding their way to Earth. The rhythm is hypnotic, soothing, perfect. Your eyelids grow heavier with each pulse of light. Your muscles release tension with each breath of flower-scented air. The meadow around you becomes harder to see, fading into darkness. But you know it is there. You trust its presence. Millions of flowers breathing in the dark. Countless small blooms continuing their work. And you, resting among them all, finally ready to release the day. The next video will be here when you wake, ready to carry you to another place of peaceful learning. But for now, there is only this moment. This breath, this softening. The flowers whisper goodnight in fragrance and dew, in the closing of petals and the settling of leaves. All of nature is settling with you, finding rest after the long work of blooming. Let go now. There is nothing left to hold. The gentle science of sleep is calling you down to dreams where gardens grow inside your mind, where you walk without worry through fields of impossible beauty, where every flower knows your name and welcomes you home. The earth holds you, the darkness embraces you, the garden releases you to rest. Good night.